Okay, I guess lipid is not something that is foreign to us, right? Most of you may have known lipids are basically fats, all right? So lipids are organic compound, they are biomolecule, and they are mostly hydrophobic. What does that mean when lipids are hydrophobic? Yes, it doesn't dissolve in water or mix well with water. Okay, it's present in various cells of the body. Okay, and it's oxidized for energy. So on top of carbohydrate, lipids are also our source of energy. Okay, and it can be converted into other essential tissue constituents, meaning that they can build up our tissue or stored as reserve fat in the adipose tissue. Okay, so compared to carbohydrates, lipids can yield twice the amount of energy. Okay, they are also a good thermal insulator. So we may joke around with our friends, those who have like higher lipids not easily get cold. Okay, so they can interfere in heat transfer between body and ambient, consequently decreasing skin surface temperature. So I've actually put in a link to a paper where they study areas with higher fat percentage have lower skin temperature, meaning that they are more stable and does not easily fluctuate when there's a change in temperature. Okay, and lipids are also our structural element for cell membrane, and they makes up the brain and also nerve tissues. So fat are necessarily bad. Okay, some of us may think we must get rid of fat and so on, but we actually require them. Okay, it's also as a protective cushion for many tissues and organs. Okay, these are the comparison between carbohydrates and lipids. So as I mentioned yesterday, carbohydrates is for rapid energy. Whenever that we require energy rapidly, so it's carbohydrate that we should go to. That's why whenever someone is feeling tired or in the symptoms of hypoglycemia, we would give them like sweet drinks rather than we give them like fried chicken, something like that, okay? Because it's carbohydrate is for rapid, but lipids is for long term, okay? Energy. Carbohydrates are soluble in water. Think of sugar. Sugar easily dissolved in water, but not lipids. Okay. All right. So because carbohydrates are easily dissolved in water, they can be easily transported throughout our body. Okay. But for lipids, although they are insoluble in water, that makes them a good constituent for our cell membrane. Okay, it doesn't dissolve in our bloodstream, for example. Okay. Okay, so how lipids are stored as energy is when we oxidize them. Okay, the oxidation of fatty acids in triacylglycerols are the principal storage form of energy. Okay, their carbon chains are in highly reduced form and the energy yield is greater than carbohydrate, which we will look into later in the digestion of lipid. Okay, so usually in exam questions, this part, the initial part, introduction lipid groups are usually in the MCQ part. So they will ask you direct questions. But for digestion of lipids are usually within the essay. Okay, essay section. Okay, so these are the main group of lipids. We have sterols, phospholipids, and also triglycerides. What are the examples of steroids or sterols that you know? Sterols. Testosterone, progesterone, and the closest one is cholesterol. Okay, that is one type of steroids. We also have phospholipids, the ones that make up our cell membrane, and also triglycerides, which are fats and oil that we may observe or see in our daily life. Okay, so this is the structure of triglycerides. So remember, it's triglyceride, so it has triglycerol, sorry, tri-fatty acid and also glycerides. So this is the glycerol backbone, and attached to it is 3 fatty acids. So that's why it's triglycerides. Okay, So the hydrolysis of triglyceride will give diglyceride and also a free fatty acid. Okay, meaning that if we add in water, okay, and allow the reaction to take place, it will cleave the bond between these fatty acids, 1, 2, 3, and this glycerol backbone. So one reaction will release one fatty acid. Okay. Okay, so these are the fatty acid chain. Okay, so these fatty acids are further classified into two. Okay, saturated and also unsaturated. Okay, can you tell me the difference between saturated and unsaturated? Double bond, branch. What else? More? Yes. In terms of which one we should consume, that would be saturated or unsaturated?
Unsaturated.
All right. Okay. So the first difference would be in terms of the carbon bonding. Okay, if you observe, saturated is a single bond between those carbons, but unsaturated, in between those carbon chain, they can form double bonds. Okay. So usually saturated are in linear structure like this one, but unsaturated, due to the double bond presence, they could be tilted or what we call as branching. Okay. And in terms of energy, sources of these saturated and also unsaturated fatty acids also differ. Okay, so for saturated fatty acids, these are the examples. Okay, in butter, beef, coconut oil. Okay, and saturated fatty acids are usually causing heart diseases. Okay. All right, and then saturated fatty acids, usually the sources will be in solid form. Okay, meaning, for example, this like butter. Okay, but for unsaturated fatty acids, we can observe them in olive oil. Okay, vegetable oil generally. Okay, so unsaturated fatty acids can be further divided into monounsaturated and polyunsaturated. Okay, so monounsaturated means it has only one double bond. Okay, remember unsaturated have double bonds. So for mono means they have only one double bond, but for polyunsaturated, two and more. Okay, so usually it's liquid at room temperature. Okay, so that is classification in terms of the number of double bonds in the unsaturated. We can also classify them according to their structure. Okay, so unsaturated can be, we have cis fatty acids, okay, or trans fatty acid, okay, trans-unsaturated fatty acid. Okay, so if you observe over here, all right, so this trans meaning they are in opposite sides. Okay, so it's usually like this. So this is a double bond. So the branching is usually like this on opposite side. But for cis is usually on the same side. Okay. Okay, we also have essential fatty acids and also non-essential fatty acids. Okay, similarly to essential amino acid and non-essential amino acid. Okay, so essential fatty acid means it cannot be synthesized in the body and need to be included in the diet. Okay, and for non-essential is something that our body can synthesize them. Okay, I think one of the good things that you need to look into here is the one of the good essential fatty acid would be the omega-3, something that we should consider taking in our diet. Okay, because it's good fatty acid. All right. So next one, we will look into phospholipids. I will go through this briefly because this is in chapter 2. Okay, so these phospholipids, which one is the phospholipid in this diagram? The kepala? This one, okay? It is in bilayer form. Bilayer means 2 layers, okay? So they have hydrophilic heads and also hydrophobic tails. So these are the phospholipids, okay? Okay, because of this phospholipid, it actually causes the properties of our cell membrane to be semi-permeable, okay? Meaning that it only allows certain molecule to go through it, okay? So lipid soluble molecule can easily pass through our phospholipid bilayer, okay? But other molecules may require channel, channel transport protein, okay, to go through it. Okay, that is for phospholipid. Okay, next one will be steroids. Okay, I think the stigma that we have on steroids is basically drugs, right? It's associated with drugs. Okay, but not all steroids are bad. Okay, we actually have steroids in our body or in the form of sterols. Okay, so one of the examples just now given was testosterone, progesterone. So most of our hormones are made up from steroids or in the group of steroids. All right. Even cholesterol in our body in the steroid group. Okay. So these steroids, one thing that you must observe over here, they have similar structure, which is this ABCD structure. Okay. If you observe, okay, for this one, cortisol, similar structure ABCD. This one is aldosterone. So this one. So A, B, C, D, A, B, C, D, A, B, C, D. So this what makes this group of hormone falls under the category of steroids. Okay. Okay, so one of the famous example of steroid will be cholesterol. Okay, these are usually present in mammals. All right. So cholesterol, although we observe it or we always identify it as something that is bad, okay, causing heart disease, for example, or heart attack. But we actually need cholesterol in our cell membrane to maintain the fluidity of the cell membrane. Okay, so most of the cholesterol in the blood are surrounded by layer of phospholipids. For example, the low density lipoprotein. Okay, and high density lipoprotein tend to carry excess cholesterol to the liver to be processed. Okay, we will look into this later lipoprotein here. Which one is good? Which one is bad? All right. Okay. So humans are also synthesizing steroids, or what we call as anabolic steroids, usually taken by those who are building up their muscles. Okay. What these anabolic steroids do, they will increase the rate of transcription of genes which encode actin and myosin proteins. Okay, meaning that these steroids enhance the development of skeletal muscle. Okay, because actin and myosin is part of the skeletal muscle, so this steroid enhance that. But although they have sort of a good effect on the skeletal muscles, building them, but these anabolic steroids are proven to be harmful on liver, our heart, and also kidney. Okay, they can also inhibit production of reproductive system hormones, okay, which can lead to infertility. Okay, and if females are taking anabolic steroids, these steroids are usually the variation of testosterone, meaning that female can develop male characteristics. Okay, there are 2 papers here you can read on later. What are the effect of these anabolic steroids on behavior, for example, and the side effects. All right. So that's generally on the types of the introduction, okay, and also the lipid groups. Okay, next would be lipoprotein. Okay, so when I said lipoprotein, what do you imagine? It's a combination of what? Protein and lipid. So lipo is the lipid, so protein is from protein, okay? So because usually lipids or cholesterol are insoluble in water, they need a carrier to be transported. So how lipids or cholesterols are transported in our body is by attaching them to proteins. Okay, so proteins can be transported. So mostly cholesterols, one type of lipid is carried as lipoproteins. Okay, so lipoprotein has 4 types, which are chylomicron, very low density lipoprotein, low density lipoprotein, and high density lipoprotein. If you're taking clinical biochem later, you will see this a lot more later. Okay, because these are one of the sources of diseases. Okay, so what makes this lipoprotein differ from each other is their constituents, meaning that what makes up them. Okay, so if you observe over here, this is chylomicron. VLDL is a very low density lipoprotein. So this is low density lipoprotein and high density lipoprotein. In certain papers, scientists classify as well as things like intermediate density lipoprotein. So there's so many more, but these are the general ones. Okay, so the blue color here is the phospholipids. Okay, within the phospholipid would be the insoluble triglycerides. Okay, and then we have cholesterol in between, the yellow colored ones, and also proteins embedded to them to transport them. Okay, so based on this figure, which one would you think is bad for us? Chylomicron or which one? LDL? HDL?
HDL.
HDL. Okay, why?
It's a good question.
Yes, Miss Lee.
It bring away all this. It bring away all the unwanted fat from our body.
Yes.
It's like transportation.
Yes. So LDL and HDL contains a lot of cholesterol in between. Okay. So high amount or excess amount of LDL and HDL are causing clog in our bloodstream. Okay, blood vessels. But chylomicron and VLDL are actually good lipoprotein. Okay. Okay, so you can read on further. I'm not sure if I attach the paper here, but you can look into later what are the difference. For example, chylomicron, they have the largest diameter. Okay, this is diameter, this is density. So chylomicron has the largest diameter, but it has the lowest density. Okay, because it carries a small amount of cholesterol. Okay, compared to small LDL, for example, or HDL, they have larger density than the chylomicron. Okay, so this density is contributed by the amount of cholesterol that are carried with them. Okay, so this is the pathway of these lipoproteins. Okay, for example, over here, this VLDL. Okay, let's take an example. VLDL over here. Okay, so VLDL will carry the dietary cholesterol that we have eaten, for example, that has been processed by our liver. So VLDL will carry them through our blood vessel or capillaries and transport them to our fat tissue and muscle, for example. Okay, so different pathway or destination will use different type of lipoprotein. Okay, for example, in the extrahepatic tissue, okay, meaning that outside of the liver, LDL is used as the means of transport to transport these fatty acids. Okay. Okay. For example, over here from the intestine, if directly go to the adipose tissue and muscle, our body will use chylomicrons. Okay. So later on, if you would like to study this, you can do a summary, okay, from where to where. Okay, so the starting point. Oh. Okay, for example, we have starting point and end point. Okay, so you can write in what are the lipoprotein that is involved. For example, starting point, intestine or alimentary system. Okay, digestive system. Sorry. End point would be adipose tissue, muscle. Lipoprotein would be chylomicron. Okay, do a summary of this so that it's easier for you to memorize this. Okay. All right. Okay, so this is a bit about chylomicrons, which I've mentioned just now. Okay, so these are the functions of it. Okay, for example, again, chylomicron site of synthesis will be intestine, so to transport the dietary lipids from intestine to peripheral tissues. Okay, okay, I think I've made mistake just now. That's a very small mistake. So transferase is not transferring them, but it's transfer between groups. Okay, translocase baru transfer between two places. Transferase is transfer between group. So meaning that acyl-CoA and carnitine. So there's a transfer between acyl-CoA. Acyl to CoA is detached. So attach carnitine. So transferring carnitine to acyl and removing CoA. Okay. So translocase from within this between outer mitochondrial membrane and inner. So locate it back. Okay, carnitine into inside, acyl carnitine inside, carnitine outside. So whenever you want to study, try to remember there's 4 enzymes like this. 1, 2, 3, 4, and we have 3 layers. Okay, 1, 2, 3 layers. Okay, 4 lah, 4 layer. 1, 2, 3, 4. Okay. The first thing must activate first. Okay, and then must transport inside. So activate is catalyzed by synthetase. Okay, and then we have to transfer the group, transferase, and then locate them inside, translocase. Okay, once inside must be converted back into acyl-CoA. Okay. Okay. So let's look into beta oxidation. So just now is only activation and transport into the mitochondria. So now inside we want to use the fatty acid for energy. Okay, that is beta oxidation. So generally it means a series of reaction. So we have a series of reaction that cleaves carbon atoms 2 at a time from the carboxyl end of a fatty acid. Okay. Okay, so in this beta oxidation, it will start with this fatty acyl-CoA. Okay, it's the same thing as acyl-CoA, but certain textbook use fatty acyl-CoA, but it's actually CoA, acyl-CoA. Okay, so this is the structure. This is the CoA group. So this CoA is bound to this acyl group. Okay, this one is the acyl group. Okay, this okay, this is the CoA group. This is the acyl group. This is the thioester bond, S. Okay. Okay. So if we start with this acyl-CoA, so the first reaction would be oxidation. So from acyl-CoA, it will turn into enoyl-CoA. Okay. So this oxidation is between alpha and beta carbon. Okay, so ni CoA. This is the thioester. So this is carbonyl group. So the next carbon to the carbonyl group is the alpha carbon. The next to it is beta. So it's the oxidation between these two, alpha and beta, and they will form double bond. So that is oxidation. Okay. The next reaction will be hydration. Okay. So because it's hydration, meaning there's addition of hydration. Water, hydrate, kan minum air. So hydration will be addition of water. So it's catalyzed by hydratase. Okay. And form beta hydroxyl acyl CoA. Okay. So from this molecule, beta hydroxyl acyl CoA, it will undergo again oxidation and form beta keto acyl CoA. Okay, and this one will be cleaved by this thiolase or this reaction are called as thiolytic cleavage and form acetyl CoA. Okay, let us set the ground rule for this one. Okay, for this beta oxidation, what you need to remember is these 4 steps. Okay, starts with oxidation, hydration, and then oxidation back and thiolytic cleavage. Okay, so it has 4 reactions. Initial molecule is acyl CoA. Final product is acetyl CoA. Okay, let's set to that. At least remember that. Okay, if you can remember all these enzyme, that's a bonus. But the ground rule will be starting point acyl CoA, and then 4 reaction: oxidation, hydration, oxidation, and thiolytic cleavage. And then endpoint acetyl CoA. Okay. Okay, so that is generally for beta oxidation. So remember in beta oxidation, 2 carbons are cleaved at a time. So remember the free fatty acid chain that we have. This one. So this one is carbon, 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 carbon. So it will cleave 2 at a time. Okay. Okay. 2 carbon at a time. And every time it cleave 2 carbon at a time, that is actually the acetyl CoA. Okay, if you see the structure of acetyl CoA, this is carbon 1, this is carbon 2. Here. So that is 2 carbon at a time. So every cycle, so this beta oxidation will take place until the end of the carbon that the fatty acid has. Okay, so this is the fatty acid chain. Or now it is in the form of acyl CoA. So this is carbon, 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 carbon. So one reaction of beta oxidation will keep here. So one acetyl CoA is formed. And then this whole chain will undergo beta oxidation, the next beta oxidation. So it will keep here. So another acetyl CoA is formed. So acetyl CoA number 1, acetyl CoA number 2. So this remaining undergo the next beta oxidation. So it will keep here. So another acetyl CoA. Okay, until the last. Okay, until we get the last acetyl CoA. Okay, that is why a 60 carbon fatty acid can produce 8 acetyl CoA because 2 carbon is cleaved at a time. Okay, so these are the details, okay, for each reaction. So oxidation is by FAD. Hydration is catalyzed by hydratase. Okay, you can highlight on that. And the next oxidation is catalyzed by dehydrogenase, but this one requires NAD. So earlier one is FAD, the next oxidation is NAD+. And finally is thiolysis, okay, or the thiolytic cleavage, the same thing. and catalyzed by thiolase. Okay, so in different textbook, they may have different representation of beta oxidation. You can look into later on which one helps you to understand better. Okay, so in biochemistry, the main highlight would be this acetyl-CoA. Acetyl-CoA. Not only fatty acid will produce acetyl-CoA, later on in carbohydrate metabolism, you will also found acetyl-CoA. Okay, even in protein, not sure. Okay, but this acetyl-CoA actually links between lipid, carbohydrate, and later on. Okay, so this one is something that you need to take on to remember. Okay, so acetyl-CoA can be oxidized into carbon dioxide and also water via the citric acid cycle, which we will look into tomorrow. Carbohydrate metabolism. Okay, yang lain ni tak apalah. Okay, so this is what I mean by 2, 2, 2, 2. Okay, if the carbon fatty acid, the carbons in the fatty acid is 12, so the number of acetyl-CoA formed would be 6. Okay, so the cycle would be 5. This is how you calculate it. Okay, if the carbon in the fatty acid is 14, then the cycle, the number of acetyl-CoA formed would be 7. Okay, because 1 acetyl-CoA, 2 carbon. Okay, and the cycle will be 6. Okay, so just now I mentioned you, we have 6 things to remember. Initial is the acid-CoA, and then